Yes, welcome everybody. We're going to get started with our webinar. So today we are going to have, we're going to start with a brief introduction to the Allen Institute and where you can find our resources for educators. And then Graham is going to introduce you to some of our selected resources that are available at allencell.org, which is our cell biology research division. And then I'm going to take you through an interactive demonstration of our featured lesson for today, which is called Mitosis and Microscopy. You can find a link to this on the login page where you clicked the link to join this Zoom meeting, as well as at alleninstitute.org/learn. And then we'll have a quick summary and recap, and then we're going to have a brief careers in STEM session. And this is primarily for the benefit of those of you who are joining us for Washington State Continuing Education clock hours. And we'll conclude with Q&A. So again, at any point, please put questions in that Q&A panel, not the chat panel. Please use the chat panel to talk to each other. And then the Q&A, we have some folks who are in the Q&A who will be answering those for you, or they'll save them for me and Graham for the Q&A session, or they'll hop in and let us know if there's anything that needs immediate clarification. So again, please rename yourself to your full name in Zoom if you can. If you're having trouble, that's fine. Just send Dukes or all panelists a direct chat message and we'll get you signed in. And this is especially important if you're requesting that clock hour credit. And the full instructions for how to get your credit are going to be at the link that you used to sign into this webinar. So briefly, a little bit about me and Graham. We will talk more about ourselves in the career session at the end of this webinar. So Graham is the director of the animated cell team at the Allen Institute for Cell Science. He came to the Allen Institute from the California Institute for Quantitative Biosciences at the University of California, San Francisco. He received his PhD in biology in 2011 from the Scripps Research Institute in La Jolla, California, where he worked in Art Olson's molecular graphics lab, and he's also a certified medical illustrator. And my name is Caitlin. I'm the training and outreach specialist at the Allen Institute. I received my PhD in neuroscience from the University of Washington. And at the Allen Institute, I work with our brain and cell science divisions to train scientists and educators like you to use our open resources for research and for teaching. So a little
really
bit
quick,
about,
Caitlin.
yes.
Before we go much further, I see a message in chat that someone can't hear anything yet. So I want to make sure that our other attendees can hear things. So if you could really quickly, everyone, just in chat, hit a +1 if you can hear us right now. That would be great. Great. Okay. So it may be an isolated issue and we will work on that separately. All right. Thank you, Caitlin. Go ahead.
Thank you for letting me know. So the Allen Institute is a biological sciences nonprofit. We're located in Seattle with focused research areas in neuroscience, cell biology, and immunology. And we also support cutting edge research across the world through the Paul G. Allen Frontiers Group. Across our institutes, our scientific process is focused on tackling complex, broad, hard problems in the fundamental biological sciences. And our approach is guided by these three core principles: big science, team science, and open science, which I'll return to in a moment. Our work generates data, knowledge, and analysis tools, all of which we share publicly for anyone to use in their work. And to date, we've collected about 8 petabytes of data. That's about 1.7 million DVDs worth. So our core principles are team science, big science, and open science. The most relevant one for you today is open science. All of the resources that we generate through our work are available online at brain-map.org and allencell.org. So getting started for educators with this massive amount of data, there are 2 main places to start. alleninstitute.org/learn. You'll find lesson plans, posters for when you're back in school, and this includes resources for both neuroscience and cell biology. And then if you head over to allencell.org/educational-resources, you'll find more links to teaching resources specific to cell biology. And a link to the featured lesson is available, and we'll link it in the chat. So most of our resources were developed with research applications in mind, but they can be used in classrooms to provide students with a genuine research experience without access to lab facilities that might not be available in all schools or when you're not in your school, when you're distance learning. We recognize that high-speed internet is also far from universal, but for many students who are learning from home right now, virtual experiments can provide a bridge to continuing their science education when you can't get into a lab in person. And even when you're back in your classroom, we hope that our open data can also be a resource for studying human cells and the brain by providing access to types of data that typical high school and some college teaching labs don't have the facilities or time to collect. So I'm about to hand it over to Graham to start talking about the Allen Cell resources. Again, if you haven't already done so, please remember to rename yourself in the participant panel. Many thanks to Irina, Dukes, and Christina, who are going to be helping out with answering questions. They'll be answering some of your questions directly and sharing some questions with us. We encourage you to chat with each other and to introduce yourself using the chat panel. So please go ahead and tell us who you are, what institution you're coming from, and what classes you teach. And then share any questions for us in the Q&A panel. So, Graham?
Thank you, Caitlin. Can you see my screen okay? Great. So I want to thank everyone for joining us. And on our website, allencell.org, which is the home of our scientific resources, we have a newer page called Educational Resources. And you can currently find this under the Animated Cell tab in the top menu up here. That page consolidates links and describes other pages on allencell.org that have been found useful for teaching. The first thing you'll find on the page after the overview section is one of our more complete and ready to use resources, which is a link to a virtual lab published in a teaching journal called Course Source. And in that Course Source section, there's also a link to a distance learning webinar we recorded and posted last month. The second half of that webinar was presented by a college professor from Washington State University, Dr. Eric Sheldon. And in it, he gives an overview of the goals of the Course Source virtual lab that he developed with the co-authors, Erica Alfredal and me. And he also gives a tutorial which has a walkthrough of how to use the pertinent online resources you'll find on the rest of the website that are required for the course, as well as how to evaluate the students' results. Further down on the page, we have videos from Eric and two other teachers, Tom Martinez and Carlos Golier, who came to the ASCB conference, which stands for the American Society for Cell Biology, to give presentations describing how they use various pages, data, and tools that are available on allencell.org in their own classrooms. And you can watch the videos on that page to find out more. And in the next section down, we have provided links to the various pages that those three teachers and many other teachers have told us they use in different ways. So if you capture nothing else from my part of the webinar, just remember that there's an educational resources page on the Allen Institute for Cell Science scientific website, which is allencell.org, that will give you access to everything you're going to see and learn throughout the rest of the presentation today. The page has all the links you'll hear about because it also includes a link to the broader resources that Caitlin is describing today on our parent website, alleninstitute.org. And that link contains, for example, biology posters you can hang in your classroom and a set of growing resources that would be useful for teaching neurobiology that stem from resources generated in the Allen Institute for Brain Science. So in a few minutes, I want to show you what each of these tools offers. And of course, for our main event, Caitlin's going to describe a detailed tutorial of how to use the 3D Cell Viewer among the other tools that are on the Allen Institute website. But first, I want to give you a brief overview of the research we conduct at the Allen Institute for Cell Science to help you understand where this data comes from, why we gather it, and what will be available in the future. And this will take about 10 minutes. I want to overview a major goal of our Cell Science Institute, which boils down to integrating the structural anatomy of cells that we can see in our microscopes with other data to be able to infer and learn the physiology of cells. I want to quickly highlight some of the data and tools we currently provide online, while also describing some of the more sophisticated tools that we hope to make available in the future. For most of our image data, we label only 3 structures at a time to minimize perturbation. What that means is that we use optical microscopy, light microscopy, and I'm going to step through the details of what that means. But we consistently dye the DNA and the cell membrane of all of our cells, and then we, one at a time, in different experiments, label various organelle structures such as actin, microtubules, mitochondria, or aspects of the nucleus with endogenous GFP tags. So we use fluorescence microscopy to collect this data. And so we're only seeing one thing at a time, mitochondria, microtubules, and we want to find ways to bring those back together to get a more realistic representation of what a whole cell looks like with all of its parts inside. For any new research, the first step is observation. To begin to understand how cells function, we first need to be able to see them and describe all the components inside of them. At the Allen Institute for Cell Science, we dive deep into this first step of observation by building 3D atlases of whole cells. And a second ago, you saw some of our raw data spinning around, collected directly from the microscopes with a field of cells butted up against one another. And here I'm showing a rotation of 2 cells that have been segmented, where their structures have been outlined from that data. And we can see one mitotic cell next to one interphase cell with recognizable mitotic structures of the chromosomes being pulled apart by the spindles in the lower right here. So why are we doing this? A lot of you in this audience can look at this image and tell me with a high degree of accuracy which 2 cells are most likely to divide within the next couple of hours, but can anyone look at the other cells in this image and tell me when you think each one of those cells is likely to divide or to do something else like migrate or differentiate or apoptose itself? And what if I gave you the time lapse, or if I let you watch 20 other time series movies of the same cell line under the same conditions, where we're watching microtubules pull the DNA apart within the bounds of the plasma membrane in pink? Would you be able to then predict when each cell is likely to divide? Or if you've been studying this for 20 years, could you do that within an accuracy range of, let's say, 4 hours? If we layered on other biological data, such as spatial gene expression data, how many mRNAs are where, or what's the proteome? Or if we saw other structures like the centrosomes, could that improve your accuracy? Could you predict any given cell to within an hour? And could a computer vision algorithm do any better? So at the Institute, we believe that by observing and quantifying behaviors like the ones you're seeing on your screen right now, we can find patterns to help crack the code of cellular complexity. And ultimately, we want to do more than that. We don't want to just predict what cells are going to do. We want to work with the cell biology community to help connect these predictions to mechanistic understanding. So what are the underlying molecular mechanics that stem from 50 years of knowledge from reductionist biology that are letting cells exhibit these phenomena? To address these types of questions rigorously, we need a lot of reproducible data, and you'll see soon how we can repurpose that data for education. So this slide is just providing a pretty overwhelming overview of our imaging pipeline that I'm going to use as a reference map for the rest of the talk. We'll simplify it a bit and just begin with the cells on the left. Our model system is the human-induced pluripotent stem cell, which has a variety of advantages you can see in the bullet list. So what this means is a biopsy was taken from a donor several years ago at another institute, and the fibroblasts in that sample were tricked by pouring certain chemicals on them into turning back into a stem cell-like state. And that gives us the advantage of having human cells with stable genomes that can be grown in culture. They replicate. There's tons of genomics information available from various precision medicine efforts going on around the world right now. But of course, a major reason we use them is because they can differentiate into a number of cell types for use in the lab. So we can pour other chemicals on them and trick them into growing into heart cell precursors or neuron precursors, liver, muscle, etc. At the Institute, we currently study cells in both their undifferentiated states and as they turn into cardiomyocytes. And if you differentiate the cells into heart cell precursors, they eventually beat beautifully in a dish, and we can visualize any of the structures we've labeled with green fluorescent protein or other fluorescent proteins at higher resolution in the living tissue. You can see in the bottom images. So in the top, you're just looking through something like a dissecting microscope, and this was just recorded with an iPhone being held up to the objective lenses. To get lots of data, we've developed a mostly automated pipeline to maximize reproducibility and efficiency. We want to visualize our cells in a semi-automated manner, so we grow them on 96-well plates that look like this, where the individual cells settle and begin to grow and divide into colonies that look like this at low magnification. And here we've used an edge detection algorithm to identify and color code the colony boundaries. And if we zoom in onto a colony, we of course find that it looks practically transparent. So our human-induced pluripotent stem cells look like this at higher magnification and white light. And to make a component pop out from the background, we use fluorescence microscopy on cells that have had one structure endogenously tagged with fluorescence proteins at a time. So I'll explain what that means. And on the left side of the screen here, we're looking at a cross-section of one of our cells. Again, our cells grow in these flat colonies in these dishes or in these plates. And on the bottom, the cell is flat because there's microgel that it's sitting on. And there are neighboring cells that would be adherent to the tight junctions at the tops of this cell. We have a large nucleus, and of course the genetic information is inside. And if we just isolate one gene as this orange sausage here, the idea is that through native proteins are generated through the normal processes of transcription and translation as the cells are just sitting there growing and dividing. And those proteins localize to their appropriate compartment, various organelle systems through a variety of different mechanisms. In this case, we're looking at how the proteins end up on the surface of the outer membrane of a mitochondria. But if we look at those cells under a microscope with just white light, we rarely get the resolution and contrast we need to distinguish the structures from the background in a useful manner. So we use technologies like CRISPR-Cas9 to take that exact same DNA sequence, and in either the mother or father genome, we insert a tag for a fluorescent protein such as GFP or a variety of others. And that results in producing a protein that has a light emitting tag, a different protein stuck onto it somewhere. And we work to find where we can put these proteins with perturbing the cell as little as possible. So the proteins are expressed at their native rates, and some of them come from one of the parental genes and some come from the other. So you end up with a mix. In this case, about 50% of them have little GFPs. And when you shine something like blue laser light on it, those GFPs emit green light, which we can see in the microscope, and it looks something like this. And if you superimpose that onto the background, you can start to pull out the signal from the noise. This gives us beautiful data, which looks like this for a cell line where the nuclear lamina was made visible by tagging lamin B protein, which again, just on the inside of the nuclear envelope and on the outside of the bulk of the DNA, there is a protein mesh that stabilizes the nucleus. That's what we're looking at here with this bright glowing outline. And you can see cell division events occurring, et cetera. We collect this data in 3 dimensions, and we have a growing number of different types of microscopes, but our initial workhorse that's been able to allow us to do this robotically and automatically has been the spinning disc confocal microscope. And these are just some examples. These are Z-stack movies, so they might look like a time series, but we're just starting at the bottom of the cells in their plates and scanning through space up to the tops of the cells. These are just various different organelle systems that we've tagged. You can find out more details about those on the cell catalog, and we'll post links to those also in the follow-up email. But these are just 18 of 30 different lines that we have available for use in the lab. People are doing research out there. A number of organizations around the world have started using lines from this cell catalog to study systems in basic cell biology, developmental biology, and in this case, we're outlining various human diseases that different labs and companies are studying. And I just show this to say, for a lot of students, this type of connection to human impact can often be motivational for certain learners. We appreciate that Paul Allen has funded our institute to do basic cell biology, and that's really difficult funding to come across, and it's critically important. But because our cell lines are available, they can also be useful in learning more about diseases. So now we're going to go over some of the tools we use to provide access to these data and integrative whole cell models, which are pertinent to different types of teaching exercises. And I'll try to do this more in the context of things that you can use in the classroom. So in the beginning, I told you we collect, and I just showed you a slide with 18 different cell lines. So we light up the DNA with dye, we light up the cell membrane with dye, and then we light up one structure at a time. We do have some more sophisticated experiments where we'll do 2 structures at a time, possibly even 3 in the future. But it's really challenging on the cells, and shining that much laser on the cells is harmful to them. So we have various reasons for doing 1 at a time, and we need to figure out how to piece those together to these whole cell models. Our automated microscopes collect tens of thousands of self-image fields. And then the cells individual cells are segmented from those fields where they're stuck to one another in these monolayer colonies. And I'm showing here just a sample of cells that have been segmented from those fields. And when we zoom out, you can appreciate how many there are. Our first step was simply to allow integration was to provide access to the data, easy access. We've done this through a variety of different means, and some of them involve online tools, so you don't have to download any software, any data, and learn how to use it. You can do it directly in a web browser, and that's really advantageous for student exercises. And we really want to democratize access to this information, so that's what those tools are about. But we do have some more sophisticated tools, such as the one on the right, which is a desktop software currently, that you can use to see the 3-dimensionality of the cells in a much more intuitive manner. So one of our most recent tools that's been out for about 1.5 years and it's being fine-tuned is called the Cell Feature Explorer. And it starts out, we have tens of thousands of cells. It's an overwhelming amount of information. We want to help people filter down to the cell that they're interested in. So we do this by plotting measurements that have been taken on the cells in a graph, and each one of these dots represents a single cell in the database. And if you choose X and Y axes for certain measurements, you can get things to stand out. Like in this case, we see mitotic cells tending to stand out to the left. If you roll your mouse over them, you can see an instantaneous preview of what that cell looks like in a maximum intensity projection. And then you can select cells and build up a gallery on the right. If you click on any one of those gallery cells, it will load directly into a viewer down below on the same web page. And you get all the normal features one would expect of a volumetric viewer. This can be overwhelming to students at first, but we try to make it as intuitive as possible where they can just start clicking and dragging, or a researcher, of course, can do the same, which is who this tool is developed for. But you get the ability to modify the look of the cell, how you can modify each channel for the DNA separate from the cell membrane, separate from the structure. And you can also download the data with that tool. So that's one of our more recent tools called the Cell Feature Explorer. And Caitlin's gonna walk into a detailed version. Also, Eric's recorded webinar that I showed you is available on the page. They both use a tool called the 3D Cell Viewer. And This tool presents the data in a different manner, and it's a little bit easier to access the whole field images instead of just the individual cells. But it has all of the same functionality where you can scroll through the cells and filter them by which tags are marked. So in the top, we're looking at only alpha tubulin cells showing microtubules and TOM20 cells, which are highlighting mitochondria. And you can load the cell into the viewer. And Caitlin's going to walk you through this again, and Eric shows you specific settings that he recommends for the particular exercise in that course source video. So we want to get these single structures back together into representations of whole cells. And there's a couple different methods for that. And the first one I want to show you that's potentially used, Caitlin's actually going to show you how it can be used in educational module they've developed. So in this case, we're looking at one structure at a time, and it just got merged. We find cells out of our database and search for ones that are of similar size and shape, and then we orient them on top of one another to fit them as closely as possible. And because the data is relatively low resolution, we actually end up with pretty good superimposition. Of course, the proteins don't know about each other, they're overlapping one another, but you get an idea, is this structure towards the top of the cell or hugging the nucleus, etc. And we've built a page, which is about a 20-minute interactive scrolling movie. And at times later in the thing, the left side actually has some interactive 3D viewers that you can use to learn all of the stuff that I just told you, that's taught in a bit more of a educational manner about the type of microscopy we use at the Institute, what our cells look like in 3 dimensions, what it means to take something like this, a bright field image, and to highlight with GFP one of the particular lines inside of it. And so Caitlin will show you some more about that. And then there's one more project I want to show you, which is an assembly of a cell that has 14 different organelles in it, and it all comes from, they're all seen in a single cell. What do I mean by that? So we use a sophisticated process called label-free determination. So we took one set of data, one image experiment, which is that gray brick of noise on the left. And we have a technology where the computer has seen the relationship of fluorescent voxels to the grayscale noise of the bright field image that was collected with at the same time with the fluorescence images. And it can recognize subtle patterns. So if you were to squint your eyes at that image on the left, you might be able to sort of see the nuclear shapes, these roundish things, but the computer can squint its eyes and it can see mitochondria and microtubules, et cetera, relatively reliably. So the result of that is we can get, from our raw data, we can see the 3 structures that were labeled, the DNA, the cell membrane, and the GFP tag structure. But we can also use the label-free technology to see 7 other structures or 9 other structures reliably. And then we can model in various well-known components in relationship to those structures. And so this tool on the website, which is described by the teachers as to how to use it in Caitlin, you can sort the organelles by their functions, and then you read a nice sort of Wikipedia level description of what the function of that organelle is. And you can see the relationship of this zooming image, kind of powers of 10 image, to the organelle structure, the whole cell structure, and the protein structure on the right. So today I hope you learn from Caitlin and myself that students can use online open databases to conduct experiments virtually. And while the virtual experiments can't replace live experiments, they can introduce new data or types of data to serve as a bridge to distance learning. As a reminder, everything I presented here is available on allencell.org, educational resources page. At the top of that page is a description of how to use the course source detailed tutorial for the virtual lab. And you can find links to the other sources I presented, as well as to the links that Caitlin's going into detail now on alleninstitute.org. And later, we'll talk about questions that we might not have a chance to get to, or questions you may have in the future can be asked on forum.allencell.org. And so we have 70 people at the Institute who do all the hard work that I just presented, and I want to turn it over to Caitlin after I thank everyone at the Institute, and I want to thank everyone on the communications team for helping us put on this interactive webinar. Thank you.
All right, thank you, Graham. So now we're gonna get into the featured lesson for this webinar, which is called Mitosis and Microscopy, using the resources that Graham has just introduced. So this lesson includes worksheets, a virtual mini experiment, and instructions for how to collect the data that students will use from our website from these resources that Graham has introduced. So this lesson is linked from the registration page from the webinar, or you can go to alleninstitute.org and scroll down to where it says Mitosis and Microscopy. This lesson is intended to be easy to modify according to the level of your students and the needs of your class. You might just keep the experimental design, not use the worksheets. You might use the worksheets and look at our data, but not run the mini experiment. You might modify the experiment to more closely align with another experiment from your curriculum. But these materials are intended to give you a starting point because it's always easier to edit than to write from scratch. So first, the learning goals of this lesson. One of the core goals is for students to describe some key open questions in biology and understand the basics of fluorescent tagging, fluorescence microscopy, and this method that is commonly used in cell biology research. And one of the elements of the lesson asks students not only to identify what stage of mitosis a cell is in from these fluorescent microscopy images, but also to look at some cells that are not tagged with a structure that is directly related to mitosis, and start thinking about the fact that the rest of the cell structures continue to exist and perform functions during mitosis. And students practice this skill of being able to identify the stages of mitosis from microscopy images as opposed to illustrations, and start thinking about designing their own experiments. All right, so here's an outline of this lesson. Again, you can find the PDF of this lesson on the webinar page for this. So it starts with a pre-lesson homework, and then it has a first class session, which is questions, a worksheet that is questions that are designed for discussion. Then students complete a pre-lab assignment that's primarily a knowledge check that asks them to review how they're going to collect the data and make sure they understand some core concepts. And then in what is intended to be the second class session, they collect the data, and then either in class or as homework, complete another worksheet that guides them through reflecting on the data and analyzing the data that they've collected. Again, all of these pieces are flexible. They can be assigned entirely as homework or run entirely in class. You can modify or replace sessions as needed to fit your class and to fit your distance learning needs, but I'm gonna walk through it as written. So while you're at school, a computer lab plus paper or digital worksheets would be a great way to run this lesson, but while you are working from home, we know that students may have access to a wider range of devices or no devices at all, and that would impact how this can be run. So if students only have access to phones, we would recommend focusing just on the worksheets, the reading, and the discussion more than data collection. So you can see over on the right here that you're looking at the desktop view of the the 3D cell viewer in the Allen Cell Explorer versus looking at it on phone. And you can see that there's just not enough screen real estate on a phone to make data collection really viable. The website wasn't designed with touchscreens in mind, but you can use it on a tablet. So if students have tablets available to them, it's not as easy to use as it is on desktop, but it's definitely viable. And then for the data collection, the in-class sections of discussion and collecting the data for especially high school students were designed to be worked on in groups. But for college students, they may be able to tackle these questions independently, depending on the level of your students. All right, so if you're following along with the lesson plan, the worksheets begin on page 11. The packet up to that point includes some background information that is really helpful for you, the educator. So this is what the students are going to see. In this pre-lesson assignment, they're going to read an article on our website called 5 Things We Still Don't Know About Cells that discusses some open questions in cell biology. And it asks students to talk about surprises in the article. What did they think science knows more about in the world of cell biology than we actually do? What are some open questions that they're really excited about exploring more? They then explore the visual guide to human cells that Graham showed you. And then they start thinking about designing their own experiment. What are the applications and limitations of the methodology that they're learning about? We then get into the in-class discussion questions. These questions are a little bit more challenging and conceptual, and than the questions that are assigned for homework. So especially for high school students, we would recommend trying to get them to work together. How you pursue this portion depends on whether you're planning to run this when you're back in your classroom, whether you're going to run it remotely, and what grade level your students are at. But in particular, some asynchronous or working alone options might include getting students to work together in a Google Doc or to set up meetings on their own time to work on these questions together at a schedule that works for them. Great, and then we have a pre-lab worksheet, getting students prepared for looking at cell images, potentially for the first time, especially if you're working with more beginning level biology students. They read the experimental design, the protocol for how they're going to collect data, and they read through that integrated mitotic stem cell, which runs wholly in the browser, as it is all of the data that we are looking at in this lesson plan. Any students who have enough bandwidth to watch a YouTube video should have enough for this. The main thing here is for students to anticipate starting what they need to do to complete each step of the data collection, where they might run into trouble, and especially through reading the integrated mitotic stem cell, starting to look at how they are going to be collecting and interpreting the data. All right, so now I'm going to walk through the actual data collection. You can follow along with me on my computer. I'm going to screen share on our website, or you can do this on your own computer as well. So there's a 3-part section to this data collection. Again, all of this is in the packet. In part 1, students practice identifying the cell cycle phases using pre-sorted images from the Cell Feature Explorer. And they're starting to identify key features in these cell images that they can use to identify and sort cells. And then in part 2, they're going to flip over to the 3D cell viewer, where the labeling of what stage of mitosis that cell is in is not visible, and they're going to be asked to sort the cells themselves. They'll then also combine their data to work as a class in order to get a better sample size so they can do some more advanced statistical analysis. And then in part 3 of the experiment, they're going to try to sort cells that have a different cell structure fluorescently tagged, something that isn't directly related to mitosis. This sorting should be more difficult. Students are not going to be asked to combine or do significant quantitative analysis on this data. It's about the experience of thinking about what is happening in the rest of the cell during mitosis. All right, so virtual experiment part 1, we're going to use the Cell Feature Explorer that Graham introduced earlier. If you're following along and you want to go look at this yourself, it's at cfe.allencell.org. And the students are going to be looking at each stage of mitosis and identifying some features of the cell. So you go to cfe.allencell.org and you scroll down to the plot. Over on the left side here of the screen where it says show hide all, students should uncheck that and then just select alpha tubulin. And then this plot now is only showing cell images that have alpha tubulin as their tagged structure. And then using this drop-down menu, we're going to select interphase and mitosis. So you can see now that we have on the y-axis, we have these cells that have been labeled, and then each individual dot represents the cell, one cell. I have it set here to nuclear volume, but what's on the x-axis is not really important for this particular analysis. And a huge shout out also to Irina, who is in our Q&A answering your questions, that she is the one who did all of the labeling of these cells. So students can then click on individual cell images, and they'll appear in the gallery over on the right. You can see that they've appeared in the gallery on the right side of my screen. Lost my mouse. So they're appearing in the gallery on the right side of the screen. If you click one of those images and scroll down here, you're going to see that cell. We can look at a few different cells and start identifying features of the labeled structure. Microtubules here is in green. I've happened to have selected all interphase cells. Let me select a metaphase cell so you can actually see a cell that is in mitosis. So you can see this one, you can very clearly see those spindles have formed. All right, so now our student has taken a look through a selection of these images in different stages. And here is their sample answers. This student has identified some visual features of different stages. The DNA resolution is not clear enough that you can necessarily see individual chromosomes, but this student has identified what they're called baskets, spindles, and how their position changes. Students might struggle with prophase, prometaphase, metaphase boundaries, especially in the next step when they're trying to label cells themselves, but they should be able to roughly identify whether a cell is dividing or not, and whether it's in earlier or later stages of mitosis. All right, so the next step of data collection, Graham also demonstrated this. We're going to use the 3D cell viewer. So this is at allencell.org/3D-cell-viewer if you want to follow along. I'm going to head over there myself. So this is what you'll find when you land on that page. Use this protein tag dropdown to select alpha tubulin, and the image type dropdown to select single cell. And then you'll see that we have this grid of cells that appears. And what's important here to note is that these cells will always appear in the same order. It's probably pretty small on your screen, but if you take a look when you load up this page yourself, you'll notice each cell has a number assigned to it. So this one is AICS-12, that's cell line number 12, _6_3. And these cells are always going to appear in increasing order of their ID number. So in order to prevent your students from just collecting data on the first 25 cells over and over, we suggest saying, all right, group 1, start on cell number 1. Group 2, you're going to start on page 2, cell number 14-4, and so forth. And this is going to make sure that those students aren't just getting the same cells over and over, which is important because we're going to ask them to combine their data as a class, and we want everyone to have unique cells. So in this section of the assignment, students are asked to look at 25 individual cells, or however many numbers you want to assign your students. We have 18 on each page, or 21 on each page here of cells. And they'll mostly see interphase. They're not likely to have all 5 stages of mitosis in their sample. So when you click on any one cell, this is what you're going to see. Here's a rendering of a cell that appears to be in dividing. So here are some cells that have been pre-sorted by students. So for example, this student has identified 16 of the cells in their sample were in interphase, 4 of the cells in their sample were in prophase, and 1 each in metaphase and anaphase. And then again, we're going to ask the students to group their data based on what, as a class, so that we get a bigger sample size. So you can use Google Forms or a spreadsheet to collect this, or if you're doing it in person, you can just collect it on the whiteboard. And then each student should record these class totals. So here is sample data for a class of 28 students who were working in pairs, each pair collecting 25 cells. So these numbers are not necessarily going to be exactly what your students collect. It depends on which exact cells they collect data from. But they're going to pool it as a class and get a much bigger sample size. All right, and then in part 3 of the experiment, they're starting to look at cells that are in another stage of mitosis. So I've pulled up here some cells that are have Sec61 beta, which is a protein associated with the endoplasmic reticulum, labeled. And you can see that it's much less obvious whether this cell is dividing or not and what stage of mitosis it's in. So trying to sort the cells with a fluorescent label on a protein that isn't directly involved in mitosis, like a microtubule, should be more difficult. The goal is to help the students learn that understanding what other cell structures are doing during mitosis is also important and worth studying. And here is the data from our sample student. They have probably overestimated the number of cells that are in metaphase and anaphase here, but they are starting to get the idea of studying these other cell structures, and that's what's important here. And then the lesson concludes with a series of questions that ask students to analyze the data graphically and arithmetically and to interpret those analyses. For students learning statistics, it helps reinforce the concept of small versus large sample sizes as well, because they're going to look at their own data, 25 cells, versus the class data, potentially hundreds of cells. They're also asked to design another experiment that applies the type of data that they just used. And these questions can either be done alone or in small groups. If they've been working together to collect data, they might continue working together. We also have at the end of the packet some above and beyond questions that are potentially for extra credit or highly motivated students. They're a little more sophisticated scientifically, a little more challenging. The first few questions ask students to think about applications of computer science to biology. These are conceptual only, no coding. And some of them ask them to make inferences about mitosis and cell properties. Students who are interested in pursuing an independent project should start here, and then they can continue exploring our data and our open source software. So what's next for students after this? Students who are interested in or required to complete an independent research project can access and use all of our data and software tools, no permissions required from us. Our citation policies are listed on our website. And of course, we love to hear about projects, so please let us know what you do. Another approach is to practice reading scientific papers and comparing them to our data. On allencell.org, in the drop-down menu all the way over on the far right, one of the options is for a list of all of the Allen Cell publications, which use data that can be found on our site. For students who are completing non-research projects about sharing science and health information, such as IB projects, our data and findings can be used to support outreach efforts as well. So in conclusion, I just wanted to briefly recap those learning goals. Students have gotten an exposure to some of the open questions in cell biology, and they've learned a key tool for current research in cell biology. And they understand not only the process of studying mitosis with fluorescent microscopy, but also why it's useful. Students also gain some practical skills in interpreting image data and designing some mini experiments. So quick summary, students can use our open online databases to conduct experiments virtually. They're exposed to data they might not otherwise be able to collect or access. And while virtual experiments can't replace the experience of working in a lab and doing a live experiment, they can introduce new types of data and serve as a bridge for while you're distance learning. You can find this lesson and more resources for educators at alleninstitute.org/learn. You can find direct links to data resources and additional material for educators at allencell.org/educational-resources. And from the page where you registered for this webinar, we will be posting upcoming webinars in this series. Please stay tuned. We'll also send you an email to let you know when those are scheduled and registration is live. So finally, thank you to the Allen Institute's founder, Paul J. Allen, for his vision, encouragement, and support. Again, please keep posting those questions in the Q&A panel. Our Q&A team is gonna continue tackling those and share some of them with us in our Q&A session, which will come right after we talk about careers in STEM. This portion of our presentation is primarily for those of you who are participating for Washington Science Teachers Association credit. Graham and I are just gonna briefly talk about our career paths and education and background in science. So I'm gonna go first. So I attended Pomona College, where I received a BA in neuroscience. I started college thinking I would study animal behavior. Specifically, I was interested in human-animal interactions related to climate change and how shifting patterns of animal behavior as a result of climate change can impact their interactions with humans. And I thought I would be a biology major. But because I was interested in animal behavior, I thought I should learn about the brain. And I took a neuroscience class and I ended up loving it. I still love animals, and I still love learning about animal behavior. Obviously, I'm not a fan of climate change, but I do find it interesting. And then ironically, I never studied neuroscience in animals. I worked with humans for all of my research. I also minored in psychology by accident because I took a bunch of psychology classes for my interest in cognitive neuroscience, and I minored in theater. Then I went to grad school at the University of Washington, where I received a PhD also in neuroscience. I picked the program at the University of Washington because it's an interdisciplinary umbrella program, not a department. It actually has faculty members who are members of this program from about 28 different departments across UW, ranging all the way from cellular and molecular neuroscience up to cognitive neuroscience working with humans, which is where I was. And then I also ended up associated with some neural engineering projects because the technology I used for my research, which is called electrocorticography, has applications for prosthetics. But my research was on functional connectivity patterns in the human brain, which is how activity in different regions is linked to each other. And it's thought to basically measure how information flows through the brain and how these patterns change. They vary both spontaneously over hours to days to lifetime, and directly as a result of learning a new skill. So my research contributed to understanding how skill acquisition changes patterns of connectivity and activity, which can be used to help prosthetics only listen to the signal of a user that's trying to move it, instead of the noise that comes from natural variation in these patterns. I also got 2 graduate certificates, which is basically a minor for graduate students at UW. So I did computational neuroscience and neural engineering. It was very relevant to my research. And I also did the Science, Technology, and Society certificate to get some formal learning on how scientists are viewed by society and how we can serve our community. I'm especially interested in how science and scientists are portrayed in fiction, especially TV, movies, books, and on stage, because I have an interest in theater, which is extremely relevant right now. So I mentioned at the beginning of this presentation that I'm the training and outreach specialist at the Allen Institute. I like to use this chart to describe my job. So training and outreach, when I say training, I mean usually materials and sessions that help people who know what resources they're interested from us learn how to use them. And for outreach, I mean helping scientists and educators like you learn about the breadth of resources we have available and help you figure out which ones are the right fit for your research or your class. So this would be a training session, and if you're interested in learning about other resources that we have to offer, please get in touch. And then I have on the x-axis here, scientists and educators. So these are not mutually exclusive groups by any means. When I say scientists in this context, I mean people who are primarily using our resources for research. And when I say educators, I mean people who are primarily using them as teaching tools. Most people are both to some degree. But most people fall more strongly one way or the other. And today we are talking about educational applications. So you're in the educator and cell science category. I do also sometimes work with the Allen Institute for Immunology and the Paul G. Allen Frontiers Group, but most of my work is related to either the Allen Institute for Cell Science or Brain Science or the Allen Institute as a whole. Also, outside of my work at the Allen Institute, I volunteer at Pacific Science Center, which for those of you who are not locals is our main science museum. They have a volunteer program that is specifically for scientists where I've been volunteering for 6 years, I think 6 years. Sometimes I volunteer at a monthly event called Meet a Scientist, which is for kids and families to get to meet scientists from different disciplines. They are running this event virtually right now, so if your students are interested, you can find the information on their website. I haven't appeared in it yet, but it is happening. And my favorite thing that I do with them is an annual event in their evening talk series where I talk about the neuroscience of different Halloween-related creatures like zombies or werewolves. Or this photo here was from when I talked about the neuroscience of Frankenstein. So Graham, I will turn it over to you.
Thank you, Caitlin. Okay, so I began my career as an artist, and I later went on to graduate school part 1 to become a medical illustrator. And I bring this up today. So these are just images showing several of the pieces I generated in traditional media, watercolor in the upper left and charcoal in the other 3 images as portfolio pieces to get into this program. But I brought this up today because I've recently been noticing an interesting parallel between the goals of medical illustration and one of the major goals at our institute in modern cell biology. And that is about assembling disparate types of data into comprehensive representations of the subject matter. So in this case, on the left, I've described the salivary glands as an illustration, and this is all based on dissections we were doing on cadavers in the lab with the med school students. And you can't just saw a human head in half and see through the tongue to see where the teeth are and the salivary glands are. You have to pick your way through, taking visual notes along the way, and then go back to the studio to create a comprehensive assembly of all those bits of information into this final piece, which was done in pen and ink on a piece of white clay-covered board. And the second image on the right is showing how the vessels move through the back of your knee. And this was again done with dissections across 2 or 3 cadavers, because you have to pull things apart and it's hard to find references from how one structure relates to another. This was done with airbrush, like you would use on a t-shirt or a van, on board with some pencil touch-up as well. So in our second year of illustration, we would actually go into the operating room, which you can see in the lower right corner, and we would go see 3 or 4, sometimes 5 or 6 of the same surgeries done on different patients over and over, and stand behind the surgeon and actually be allowed in with pencil and paper, sketch over their shoulder to create visual notes of what was going on, and again, take that back. So in this case, this is a color pencil illustration I did with some airbrush showing how to remove a cancerous section of the small intestine, and a piece showing how to insert a drug into the eye that is harmful to the liver, so they have to put it directly in the eye. The eye has a blood-brain barrier or an eye-brain barrier, so that drug can't get out of the vitreous humor of the eye and hurt the liver. I took this all across the country to the Salk Institute and started working on new illustrations for a cell biology textbook in the late 90s. And it was really fun activity because the illustrations of molecular biology and cellular biology at the time were mostly circles and triangles with reaction arrows in between them, but something called the protein data bank was being fleshed out, and we were beginning to see what the actual shapes of those circles and triangles were. So it was fun to be in that place at that time to redo an entire textbook from scratch with that new knowledge. So these are some pieces at the top of more recent versions of that textbook. I also had the chance to do a variety of animations, and this is one of the more popular pieces I did in '99 and 2000 for Science Magazine, showing a motor protein called kinesin and how it moves along a microtubule. And this is a huge collection of different types of data, including kinetic rates from biochemistry, protein structures from the protein data bank, versions of how the steps, pieces of the time of one position to the next, which were collected with electron microscopy at the time. Worked on whole cell assemblies from tomograms for a while in graduate school with the idea of being able to reconstruct models similar to what we're doing at the Institute now. And I focused primarily on a project to build molecular level of detail into components of cells and organelles. And so our main goal is to take all of that experience and to apply it at a large scale, at an industrial scale at the Allen Institute, to begin to assemble all this information into the context of our cells. So what are components like actin-mediated endocytosis? How is that going on inside of these human-induced pluripotent stem cells? And this takes a team of diverse specialists working again with that larger team of the 70+ people and all of our collaborators around the world, ranging from illustrators to game designers to biochemists to biologists and chemical biologists and graphic programmers and user experience designers, etc. So it's been a really fun opportunity to bring this career to a place at the Allen Institute where we can really find funding for this reassembly of all of our knowledge of cell biology. And that's all I have. Thanks, Caitlin.
All right, thank you everybody for attending our webinar today. We now have time for questions. So again, please go ahead and pop those questions into the Q&A box. And also, you're welcome to chat with each other using that chat feature. So you might be brainstorming some examples of, with the technological restrictions and potential inequity across your classroom in mind, how can you tackle a lesson like this one with your students while they're distance learning? And how can we promote student collaboration asynchronously to best accommodate students' needs at this time? So I'm seeing a question in the Q&A panel from Alice. Does the program tell you what the feature is tagged for? I just gave a number and label and then told us that it was for the endoplasmic reticulum. Yes, so let me go ahead and show you where you can see what protein you're looking at. So we are looking at the 3D cell viewer here. And if you click on a cell, it will show you up here in the top bar what protein is tagged, alpha tubulin in this particular case. And then you can, if you don't know necessarily what protein that, or what structure that protein is associated with, you can then go back and look it up. But it does always show at the top of the viewer here what protein is tagged in that specific cell.
I'll add to that in the, is it okay if I share my screen again real quick? In the visual guide, we tried to provide some information. Are you able to see this visual guide, Caitlin? Okay. So this is kind of a summary of all of the major types of organelles. So again, the goal is to see all of the structures in the cell and what they're doing relative to one another. And each structure at the organelle scale, like a mitochondria, has hundreds and hundreds of different types of proteins, lipid bilayers, et cetera. So we want to see the collective action of those proteins working together. So our goal is to tag proteins that, when they have an enormous GFP dangling off of them, will perturb that function in the most non-perturbing way possible. So we weren't tagging proteins to do experiments of their protein-protein interactions. We're doing it just to see the overall results of those. And so in the visual guide, some of the structures that are available do have descriptions all the way down to the protein level of what that particular protein that was tagged does. And we actually have updates coming for the rest of these, such as mitochondria. It doesn't have another level here, but it describes what the TOM20 protein does in that system. And you have illustrations to a cross-section of the whole cell where each organelle that's within the 14 different organelles, the visual guide is color-coded, and then a zoom in to a more detailed version of what that looks like. And for the ones that have a protein description, again, we'll have them all up in a couple weeks. There's another layer of zoom where you can zoom in and see the protein structures themselves. I'll just show you what that looks like for mitochondria. Oops. Oh, sorry. I'll check back and show you what one of the protein illustrations looks like in a second. But I think that answers the question that there will be a resource here where you can see the size and shape of the protein relative to the whole cell.
Great, well, you go ahead and pull that up. I will also briefly mention, for anybody who is requesting that continuing education clock hour credit, please take a look at the website for the full instructions, and specifically that we'll need you to forward us your confirmation email from the WSTA. You can request that hour at any time for 7 years, but if you want to request it now, please do that by Thursday.
I got it.
Great.
Sorry to interrupt again. So for Golgi apparatus, I'm scrolling around. You can zoom in and see the whole whole cell, and you can see what it looks like as it's going through the different phases of mitosis. Here's a more recognizable phase where you see the DNA pulling apart. And the Golgi apparatus breaks down significantly from its interface structure like this. And then all of the figures, once we complete this, we'll have a complete illustration. So you zoom into the whole cell, see an organelle scale, you zoom into the organelle, and you can see the size of the protein sitting in the bilayer and the relative size of the GFP tag, so you can get an idea for how perturbing or non-perturbing that tag might be when it's dangling off of a protein that's almost similar size. Thanks.
We do have 2 more questions for Graham. Graham, does your book, or do you have a book or site with your images? We're getting a lot of great comments and kudos for your art, and they're saying they would be good for an anatomy course.
Am I allowed to advertise my own textbook? So I've worked on that book for 4 editions. This is the 3rd edition, Cell Biology. The lead author is Tom Pollard, which you can find. And it has all of the figures inside. And they also have a digital version for it. Another good book is David Goodsell's Machinery of Life, or the Protein Data Bank itself, which has hundreds and hundreds of illustrations of interesting proteins that have some functional relevance to humans, not necessarily even in their body, but in other aspects of biology. And he's been doing one of these a month for almost 20 years now. So it's a really comprehensive resource, and that's at rcsb.org. Or easier to remember is pdb.org, standing for proteindatabank.org.
Thanks.
Thank you.
And Graham, can you tell us what your grad degree was in? Kristen missed that.
Sure. So I have a master's degree in medical and biological illustration from the loquaciously named Department of Art as Applied to Medicine at the Johns Hopkins School of Medicine. That was a 2-year program. And then I worked for about 10 years before I went back for a PhD in biophysics at Scripps Research Institute in La Jolla, California.
Thanks. I do have a couple more questions before we go, if anyone can stay. I was wondering, are your educational resources appropriate for general high school or for AP classes?
So it's going to depend on your class. This lesson plan was written for motivated students in a general biology class, potentially with modifications to the worksheets to help them with some of the more difficult questions, either eliminating those questions or providing more guidance, or at the AP level and into the early college level. The core source lesson that Graham mentioned was designed for the college level. You could, again, with some modifications to help students with those questions, run it at the high school, probably AP level as well. And we do have some resources on our website for teaching neuroscience. Those can be used, again, at the advanced or AP level in high school, but they're primarily intended for college.
And if you watch the videos of our 3 different teachers from ASCB on the educational resource pages on AlanCell.org, there was a high school teacher, Tom Martinez, who describes how he's used it even at the freshman high school level at a much broader introductory class, where he's guiding the students through things like this visual guide that I'm showing, because it has language that was written for people. It assumes that they have had basically a Bio 101 type class, but a teacher can just use the visuals and walk people through it as well.
Yeah, that's a good clarification. I was referring to the specific lesson plans, but you can use the images in a much wider range of classes. I actually have gotten a couple reports from teachers at the middle school level who are using the cell images to start introducing their students to the differences between illustrations of cells versus real images of cells.
Yeah, and you guys as teachers probably know this way better than we do, but we hear from our teachers who use this, just as Caitlin described, that it's very motivational for students to know that they're looking at a real cell. And Tom Martinez describes challenges that come from textbooks, such as he'll ask a question, what color is the nucleus and where is it in the cell? And the students will inevitably say it's blue and it's in the upper right corner of the cell, because that is the one image they saw of a cell in their textbook. He's a very good speaker, yeah. And then that will tie into the more formal resources that Caitlin's described today that can be extensions of that, of how to use it as more formal assignments for the classroom.
W. Cesario was asking earlier, and Arena answered, but I was hoping you might be able to screen share. Are these videos of the HIPS cells behavior available for educational purpose?
Yes, so I'll, just to be clear too, so Caitlin showed you some good formal resources they have on Allen Institute that can walk you through this. This website was built primarily with researchers in mind, and we've just had teachers come to us and tell us how they use these research tools in the classroom. So one of the more hardcore research pages is a thing called the cell catalog here. And you can actually come here to order cells for use in your lab, but you can drill into this to see time series movies. In this case, we're looking at a Z-stack scrolling movie. I think this is a time series here, where you can see the behavior of the structures over time, and you can see it at multiple different resolutions, for example.
W. Cesario is also asking, do you have images with these cells differentiated that students can see the molecular differences between stem cells and differentiated cells?
So the short answer is both yes, but it's not very satisfying yet. There is differentiation in the cardiomyocytes. So we're really working on this just now, meaning that it's very difficult to figure out how to capture these in the microscope, especially when they're beating. And you want a three-dimensional version because you have to capture one slice at a time while the thing is moving so rapidly. So we have a brief description of what this looks like with an image like this, which I got to under cells and biology research projects, differentiation in the cardiomyocytes. You can also just use the search function on the page for cardiomyocyte to get you to that. But we are figuring out how to automate the collection of these data, so, or of these cells, so that we will have both time series and three-dimensional structures, just like we do for the HIPS cells, eventually in interactive 3D viewers on our website. It might be a year away, it might be 2 years away. It's hard to say right now.
One of the last questions, if I have questions later as I'm implementing ideas, can I reach out to the Allen Institute to ask those questions?
Yes, so Graham is gonna point us to the forum. We do have a category on the forum specifically for education. Please feel free to contact us there. Somebody will get back to you and answer your question. And we'll be posting the Q&A session and all of the links that we talked about today in that section on the forum, where you can also see the thread from our previous webinar. You can also find more technical details, and you can ask questions about the data using any of these other categories as well. Graham, anything to add on the forum?
No, just that the forum has primarily been populated by research questions, and we would really love to get in touch with the educational community. So don't be afraid to ask anything there. We'll do our best to answer it. I just want to add too that we do have some, again, we're talking about research projects that are being repurposed for education here. We do have plans in the future for developing specific educational products. And one of those involves interactive simulation so that students will be able to adjust things like actin molecules moving around, adjust their biophysical rates to see what the consequences of that and learn actively through cause and effect. And this is probably 2 years away from you being able to use it, but there's a lot to look forward to in the future. And if you stay in touch through the means of the forum, for example, you'd be able to be updated about that, and through the outreach that Caitlin and her team provide as well.
Yeah, so we are just about out of time, but I will say I see your question, W. Cesario, about if your students have questions about the research, that you and your students can get together and make a post on the forum. We are here to answer any additional questions that you and your students might have, or as you're trying to implement this in the class, and that is the best way to get a hold of us. I will be in the forum, Graham is in the forum, lots of our other staff members are there. So again, thank you so much for being here today. I see a couple more questions have just popped up in the Q&A panel, and we will answer those on the forum. Please stay tuned for information about future webinars coming up. If you've requested WSTA credit, please email us that confirmation, and we will see you at a future event. Thank you so much for being here.
Thanks, everyone.